Thank you for the kind introduction. I would like to thank the organizing committee and awarding committee of this exciting conference. I'm honored to receive this highly honorable award. I'd like to start talking about the genome editing. Programmable nucleases are tools used for genome editing, and they recognize a sequence called target sequence and make a double-strand break at the target sequence. Once double-strand break is generated, this double-strand break can be repaired by cell's endogenous repair process, which is called non-homologous endojoining process, which is error-prone process which can lead to these insertions or deletions. So these insertions and deletions are collectively called indels. And once these indels are generated in the coding sequence of a certain gene, then the gene can be knocked out by frame shift mutation. So by making one double-strand break at a certain gene, we can knock out that gene. And in addition to knocking out a gene, we can also correct mutations in certain genes by homologous direct repair process. For this process, we need to add one more component of genome editing, like donor DNA or single-strand deoxynucleotides. So in the presence of this homologous template, this double-strand break can be repaired by another DNA repair process, which is called homologous direct repair process, which can lead to insertion of those genes and or point mutagenesis in a highly precise manner. However, this homologous direct repair process, the efficiency is pretty low as compared to NHEJ-mediated index generation, and usually the frequency is almost one-tenth of the NHEJ-mediated index generation. Usually the index frequency can be like 20 or 30% when we use ordinary good CRISPR-Cas9 system. But the HDR-mediated repair process is one-tenth, which is 2 or 3% or lower than in some cells. So this low efficiency is one of the problems when we use genome editing using homologous direct repair process. There are three classes of programmable nuclease: zinc finger nuclease, and TALEN, and CRISPR. When I started this field of genome editing, zinc finger nuclease was the only programmable nuclease that we can use. But in 2011, TALEN came out, and 2012, CRISPR came out. So now most people are using CRISPR. CRISPR are adaptive immune system found in bacteria and archaea, and they are composed of actually three components: Cas9 and CRISPR RNA and Tres RNA. And these two RNA can be combined into one RNA called single RNA, which was generated by the Emmanuel Charpentier and Jennifer Doudna. And this single RNA and Cas9, only two components are now required for CRISPR-mediated genome editing. And PAM sequence is recognized by Cas9 directly. And the rest of 20 base pair sequence is recognized by guide sequence in single guide RNA, which form Watson and Crick base pairing with the target DNA. So these 20 plus these two, so 22 base pairs are matched. Then Cas9 has a nuclear domain which is activated, and they make a double-strand break in this region, 3 base before the PAM sequence. Make a blunt double-strand break. So Cas9 is like a genomic seizures. So some people ask whether Cas9 looks like a seizures. This is the shape of Cas9 when we see the atomic force microscopy. This is the structure of DNA. This is Cas9. So they don't look like a seizures. They look like a ball or irregular shaped ball-like structure. And this Cas9 and guide RNA bind to this DNA and make a double-strand break at the target DNA. Then I'll talk about the other kinds of genome editing, which is the deamination by base change. So this is the structure of cytidine, and as you can see here, there is an amine residue. If we remove the amine residue, then cytidine becomes uridine. Uridine is recognized as thymidine by DNA polymerases. Also in adenosine, this is amine residue. This adenosine can be converted into inosine by removing this amine residue. And inosine is recognized as guanosine by DNA polymerases. So C to T conversion and A to G conversion is possible when we remove amine residue from C or A nucleotide. To induce deamination in a target-specific manner, W combined this Cas9 nickase with cytidine deaminases. And this Cas9 nickase recognizes the specific target sequence, and they make this kind of burst, and deaminase is bound to the Cas9 by this linker sequence. So deaminase cannot go not anywhere. They are combined to here, and they make C to T conversion. Similarly, W made a hypothetical deoxy deaminases. Actually, deoxyadenosine deaminase doesn't exist in nature, but they made this kind of deoxyadenosine deaminase by continuous evolution called PACE, phase-adjusted continuous evolution. And this synthetic enzyme is linked to this Cas9 and Cas9 nickase and guide RNA, and so A to G conversion can be induced. So these are collectively called base editing. Now prime editing. Prime editing is a very new genome editing technology, the most new technology in our field. And it is composed of Cas9 nickase and reverse transcriptase. And they use not single guide RNA, they use PEG RNA, which is longer than single guide RNA. And 3' end of this PEG RNA, pgRNA, PEG RNA, can bind to this region of DNA, and reverse transcriptase convert this RNA into DNA sequence. And this DNA sequence, which contains this edited sequence represented by red color, can be integrated into the gene. So this editing can be induced into this gene. So this prime editing is very new, and according to our study in preliminary studies, recent studies, this efficiency of prime editing can be improved. So, which means prime editing efficiency is in most cases limited as compared to base editing, but in some cases the efficiencies can be reached to 40% or 50%, but in many cases it's like a 10% or only 5%, but which can be improved in the future. But the strength of this prime editing is we can convert any kind of small change to the genome. So this is the biggest advantage of prime editing. So far, I have talked about the Cas9, but there's also another program nucleus in this field, which is called Cas12a or CPF1, the same term. This is the structure of Cas9, and this is the structure of CPF1. CPF1 has only one CRISPR RNA. They don't have a trace RNA. And their PAM sequence is a T-rich. The TTTV is a PAM sequence, and they make a double strand here. So so when you cannot use a Cas9 because you don't have a PAM in the target sequence, then you can search and the target sequence contains this T-rich PAM, then you can use CPF4 in that case. So if you want to use Cas9 or CPF1, you have to make first, you need to start from the cloning of one guide RNA. Then we deliver the guide RNA encoding plasmid together with the plasmid encoding Cas9 or CPF1. Then we can wait 3 days, and in 3 days after the delivery of this transient transfection of plasmid encoding these two guide RNA and this nuclease, then we harvest the cells and PCR amplify the target region using specific pair of primers, and those amplicons are subjected to deep sequencing or T7 assay. This conventional evaluation process takes about 3 days, and highly experienced student in my lab can evaluate maybe 40 or 50 guideline activities over 2 weeks. But it is highly labor-intensive work. So I wanted to improve this process. So instead of cloning one guide RNA, I made a guide RNA and target cell library, 10,000 of guide RNA and a target paired guide library. To this library, we delivered the Cas9 or CPF1. Then we harvested this target sequence, harvested genomic DNA and amplified the target sequence using PCR, using common pair of primers, and those amplicons were subjected to deep sequencing to evaluate thousands of guide RNA activities by one experiment. To make those libraries, we start from the oligonucleotides, which contains guide sequence and target sequence. And from this oligonucleotides, we make a plasmid library by Gibson assembly. Then from this plasmid library, we generate a lentiviral library. And those lentiviral library was transfused to 293 T cell to make a cell library. So each cell in this library expressed CRISPR guide RNA, and they contain corresponding target sequence. But target sequence those target sequences, nothing will happen until we introduce CPF1. So once CPF1 is introduced by transient transfection or lentiviral delivery, CPF1 binds to express guide RNA, and they make a double stranded break at those target sequences, depending on the activity of guide RNA. And this in-depth frequency in the target sequence, to measure this in-depth frequency in the target sequence, we amplified this target sequence using common pair of primers, and those amplicons are subjected to deep sequencing and data analysis. These experiments were done by two talented students in my Higon and Myungjae in my PhD student. They are now postdocs. And so far we have delivered method to evaluate thousands of guide RNA activities by one experiment. But we wanted to do something more than those doing the experiment. Then I watched TV and I saw AlphaGo, a deep learning-based computational model, win the Go game against human Go champion like Lisedor and Kazi, which suggests that deep learning is a powerful tool to powerful model to perform this Go game. Not only this Go game, maybe in many fields, deep learning shows a good performance as long as they have a lot of data. If the data is not sufficient, then the conventional machine learning programs performance of conventional machine learning programs are better. But if we have sufficient data, the performance of deep learning-based models are surprisingly good. So because we knew this, so we used deep learning to our study. So we generated 15,000 of target sequence activities of CPF activities at 15,000 at target sequences, and using this big data, using deep learning network composed of one convolution layer and 3 layers of fully connectable layers. And we finally developed a computational model called Deep CPF1. This work was done in collaboration with Seong-ro Yoon from SNU. And this is the result. This is the x-axis is a deep CPF1 activity predicted by deep CPF1, and y-axis is measured in the frequency using conventional approach. As you can see here, we observe a beautiful correlation between two values, indicating that the deep CPF1, the computational model, is a very highly accurate. But someone can say that the correlation is only 0.87%. I'm sorry, correlation is only 0.87, which is not 1. So 1.99. So maybe this is not accurate. But if you repeat the experiment in biology, this is almost maximum that you can achieve. Almost maximum. So 0.0 is almost maximum that you can achieve. And so you don't need to do any experiment. You can just type in this website, and this website you can just enter your target sequence. Then this program will do everything that you want to do. They will find the target sequence, and they will find the activity at those target sequences. And those was about the CPF1. Next, we performed the similar experiment, and we delivered the deep SPK9, a model that predicts SPK9 activity. In case of SPK9, there were many tools in our field. And among them, the best was the one 2016, which was from MIT group. And we compared our model with other models, including this Da Vinci group, and using this data published by the other groups. So our model showed best performance as compared to any of these previously developed models, including this MIT group model, indicating that our model performance is best. So if you want to use Cas9, then you can use deep SPK9 for your research. And this is the structure of a Cas9. This is the PAM interacting domain. They have a leg domain and nucleus domain. And this is the structure of Cas9. And several groups made different variants of PAM variants, which recognize different PAM sequences by making this kind of different mutations, like this mutation. There are there are several different PAM variants developed in this field. But we were confused which PAM variant we should use when we target certain sequence. So to find the answer for that question, we meticulously compared the activity of those PAM variants using our high-throughput approach. So, this is a 4 nucleotide PAM sequence, 256 N 4 nucleotide PAM sequences. So first N, second N, as is shown in the y-axis, and third and fourth nucleotides of PAM sequences on the x-axis. Out of these 256 4 nucleotide PAM sequences, PAM candidate sequences, 61%, so which is 156, can be used as PAM sequence, at least one of the PAM variants. Among the PAM variants, SPK9NG showed the widest PAM compatibility. And next to the SPK9, wild-type SPK9, and VRQR variants can be used for certain PAM variants, and XCAS9 also can be used for certain PAM sequences. So not only PAM variants, people also made several high specificity variants. So making these kind of different mutations, like mainly in the reg domain or nucleus domain. So there are so many different PAM high fidelity variants. So each paper suggests that their variants are best. So we were also confused. So we compared these variants using high throughput analysis. We observed that this is a PAM target sequence, PAM sequence is here, and this is a 20 base pair target sequence, and we generate the one base pair mismatch, and we determine whether the one base pair mismatch is tolerable or not. So if one base mismatch is not tolerable, which indicates that the variance is highly specific, then we observe that those variants are specific. So Avocastan was highly specific, and Spandocastan was least specific. When you compare the general activity and general specificity of these variants, there we observe the trade-off. So these highly specific variants show the low general activity, and this SPCAS9, which showed the greatest activity, but they showed low specificity. And I'd like to talk about our department. So this is a picture taken in our department 70 years ago. So even before Korean War, our department did an experiment, like a chymography experiment. And not only the research, so we also make some like textbook, which is for pharmacology, and this textbook is used for nationwide in our other medical schools. This is our department picture, which was taken last year. So you can tell because we are not wearing masks. So we couldn't take a picture this year because of coronavirus epidemic. And this is our previous director of our department, Kyung-soo Park, who led our department very. He did a great job as a director. And this is our lab members, and especially the Myungjae and Hee-gon and Nahe greatly contributed to the work that I presented today. I'm really honored to receive this honorable award, and and my job is to teach young scientists, so they can also. I hope that they can get this honorable award in the future. I'd like to thank my lab members again, and my collaborators, especially Professor Yoon from SNU and other collaborators. And thank you for your attention.